Welcome back, everyone. Today, we're unpacking what is honestly one of the most elegant, historically significant techniques in all of molecular biology. If you've ever wondered how scientists actually read the invisible genetic code that makes up literally all living things, well, you're in exactly the right place. In this explainer, we're going to break down the entire workflow step by step. We're going to treat the complex operation of Sanger sequencing like it's a microscopic factory assembly line. It's going to be highly accessible, totally practical, and honestly, a lot of fun. So, this method is basically our blueprint for reading the code of life, step by microscopic step. It's formally known as the Chain Termination Method. And let me tell you, it was an absolute game changer when it hit the scientific community back in 1977. You can think of it as a way to physically spell check the fundamental instructions of an organism. And hey, I know the terminology can sound a little dense at first, but the underlying logic, it's incredibly intuitive once we break it down. All right, so here's our game plan. We'll briefly touch on the method's legacy as the gold standard and then walk straight into the lab. We're going to prep our sample with PCR, mix up our microscopic reaction ingredients, see how elongation and chain termination actually work, sort it all out using capillary gel electrophoresis, and finally, read the final chromatogram exactly the way a modern researcher does. Let's get right to it. Kicking things off with Section 1, the sequencing gold standard. Let's see how this revolutionary method really built the foundation of modern genetics. You know, what's genuinely wild here is that a technique developed by Frederick Sanger in 1977 remains the absolute gold standard for validation in labs today. Sanger literally won a Nobel Prize for this. For decades, this dominated the field. It's the exact same technology that powered the monumental Human Genome Project. And yeah, sure, today we have these flashy next-generation technologies that can read whole genomes in a matter of hours. But Sanger sequencing, it is still the heavy hitter for pure precision. If a lab finds a brand new mutation with a fancy, fast sequencer, they're almost always going to double-check it using Sanger sequencing because the accuracy for targeted reads is just unmatched. Moving on to Section 2, sample preparation and PCR. Now that we know its legacy, let's start up our factory assembly line and see how we actually prep our biological sample. Okay, so before we can sequence anything, we obviously need enough material to work with, right? A single strand of DNA just isn't going to cut it. So first up is amplification. We run the sample through a process called PCR, which is essentially a molecular photocopier, churning out millions of identical copies of our specific DNA region of interest. But remember, DNA naturally exists as a double helix, that tightly bound, twisted ladder. To actually read the rungs of that ladder, we have to apply heat for denaturation. This effectively unzips the double helix into two separate single strands, and one of those single strands, that's going to serve as our master template. Which brings us to Section 3, the microscopic reaction ingredients. With our sample unzipped and ready to go, let's gather up our chemical ingredients. All right, let's look at this single tube recipe. We're tossing all these components together to create a highly controlled molecular reaction. It's kind of like getting a microscopic construction crew together. First, we've got our single-stranded template DNA. That's the blueprint. Then a DNA primer to initiate replication. Think of it as the starting block. Next is DNA polymerase, which is this incredibly fast biological machine, the builder enzyme that does the actual heavy lifting. And then, of course, we need the bricks, our normal deoxynucleotides, or ddNTPs. But here is the absolute genius of Sanger's method. We also throw in a tiny, carefully measured amount of fluorescently labeled dideoxynucleotides. We call these ddNTPs. Now, you might be wondering, what's the difference between those normal bricks and the modified ones? It all comes down to a clever little chemical feature. Standard ddNTPs possess a 3' hydroxyl group. You can just think of this as a chemical hook that allows the chain to keep growing. When the polymerase enzyme adds a normal brick, the hook is right there, ready to grab the next one. But those modified ddNTPs, they're fundamentally broken. They lack that 3' hydroxyl group, no hook. So the moment the polymerase enzyme accidentally grabs one of these modified bricks and adds it to the chain, construction hits a brick wall. Boom. It acts as a permanent stop sign that immediately terminates chain elongation. And to make this even cooler, modern Sanger sequencing uses terminator technology. Basically, each of our modified stop sign nucleotides is wearing a unique, high-visibility fluorescent vest. Adenine flashes green, cytosine flashes blue, guanine flashes yellow or sometimes black, depending on the software you're looking at, and thymine flashes red. Definitely keep these colors in the back of your mind because they are literally going to become the language we use to read the final sequence in just a minute. Let's dive right into Section 4, elongation, and chain termination. We're going to see how this builds a chain reaction that intentionally stops itself.
Okay, so here's how the core mechanism of our factory actually runs. We put our tube into a machine that cycles through different temperatures. First, denaturation. It heats up to separate the strands. Then, primer annealing. It cools just enough for our starting block primer to bind to the template. Finally, DNA synthesis. It warms back up to the perfect working temperature for the polymerase. This enzyme starts racing down the strand, grabbing normal hook bricks by the thousands to extend the chain. But remember, we sprinkled in a few of those modified stop sign bricks. Eventually, completely by random chance, the polymerase reaches out and grabs a fluorescent ddNTP instead of a normal one. The instant that happens, chain termination. Synthesis stops, dead in its tracks.
Now, the beautiful irony here is that to read a complete DNA strand, we actually have to create millions of incomplete fragments of every possible size. Since we have millions of template strands in our tube, and those stop signs are incorporated totally at random, the stopping point is different every single time. One chain might get 10 bricks in before hitting a stop sign. Another might get 11. Another might get 50 or 300. So when the reaction finishes, we don't have a tube full of complete DNA copies. Nope, we have a chaotic soup of millions of varying DNA fragments. And crucially, every single one of those fragments ends with a glowing, color-coded nucleotide that tells us exactly which letter stopped the chain. All right, Section 5, capillary gel electrophoresis sorting. We've got our chaotic soup, so let's move to the sorting phase of our assembly line. This step is just a brilliant piece of physics. We take our molecular soup and inject those fragments into a capillary gel tube. Now, this tube is ridiculously thin, literally like a strand of hair, and it's filled with a special gel polymer. Because DNA molecules naturally have a negative charge, we can apply a strong positive electrical pull at the far end of the tube. When we do that, the DNA fragments are magnetically pulled right toward it. So the electrophoresis race is on. Think of the gel matrix inside the tube like a really dense, thick jungle. If you're a tiny, short DNA fragment, you can easily slip right through the vines and branches, racing rapidly toward the positive charge. But if you're a much larger, heavier DNA fragment made up of hundreds of base pairs, you're going to get tangled up and move through that gel much, much more slowly. So it's a simple physical rule. The shorter the fragment, the faster it moves. Because of this race, the fragments exit the capillary tube in a strict, perfectly ordered single-file line based on size. The fragment that got stopped after just one single base pair, that one crosses the finish line first. A fraction of a millisecond later, the fragment stopped at 2 base pairs exits, then 3, then 4, and so on. It's incredible. We've taken that chaotic soup and organized it into a perfectly sequenced parade, differing by exactly one single nucleotide at a time. And that brings us to our final step. Section 6, reading the final chromatogram. Our products are sorted, so let's check out the final scanner to reveal our data. Let's dive right into how this scanner translates light into pure genetic information. Positioned right at the finish line of that capillary tube is a laser. As each bundle of fragments exits, the laser fires, exciting the fluorescent dye attached to that final stop sign nucleotide. Then, a super-sensitive optical detector reads the specific color of the emitted light: green, blue, yellow, or red. All of that color data is instantly fed into a computer, which continuously records the flashes of light and reconstructs it into a visual chart. And here is what the computer spits out, the final chromatogram. It looks like this long, spiky graph displaying a series of colored peaks. These peaks represent the exact DNA sequence. Each peak is one flash of light. Got a green peak? That's an adenine. The next peak is blue. Boom, cytosine. By just reading these peaks left to right, which corresponds to reading our fragments from shortest to longest, a researcher can literally just read the DNA sequence straight off the screen. It's incredibly clean and visually striking data. But we do have to remember one quick thing. The sequence we just read on the chromatogram belongs to the newly synthesized strand. Thankfully, DNA is extremely predictable. It always pairs up in specific ways, A with T and C with G. Because of this, we can easily work backward. If our chromatogram reads out A, T, G, C, T, A, we know for an absolute certainty that our original unknown template strand was T, A, C, G, A, T. Just like that, we have perfectly deduced the complementary sequence and decoded the hidden biological blueprint.
Sanger's sequencing truly is a masterpiece of biochemical engineering. It gives students and researchers the confidence to validate exactly what's happening at the molecular level. But it does leave us with an exciting question. If Sanger sequencing is our gold standard for accuracy, How might we improve it to sequence the billions of base pairs in the human genome faster? When you're looking at a genome that's 3 billion base pairs long, running one tiny capillary tube at a time becomes a pretty massive bottleneck. So think about that as we wrap up today's explainer. We've mastered the art of the perfect, highly accurate read. The next step in the scientific journey? Figuring out how to multiply this entire process by millions, all at the exact same time.